Well, here we are in week 8 of our course. This week we're talking about the reproductive systems, and I have a couple of chapters out of the Bible I want to read to you. The first one is Genesis 1, starting at chapter 27 and reading 27 and part of 28. God created mankind in his own image. In the image of God, he created them. Male and female, he created them. God blessed them and said to them, "Be fruitful and increase in number." So part of God's plan is for us to have offspring. Now I'm reading over in the Song of Songs book, also called the Songs of Solomon. And in this book, the whole book is about the importance of the loving relationship and a sexual relationship between a husband and his wife. You could read almost anywhere in the book and come to that conclusion. I'm going to read I'm going to start at number 9 in chapter 4, and then I'll just read a while so we just kind of get the idea of what's going on here. You have stolen my heart, my sweetheart, my bride. You have stolen my heart with one glance of your eyes, with one jewel of your necklace. How delightful is your love, my sweetheart, my bride. How much more pleasing is your love than wine, and the fragrance of your perfume more than any spice. Your lips drop sweetness as the honeycomb, my bride. Milk and honey are under your tongue. The fragrance of your garments is like the fragrance of Lebanon. So, like I said, I'm going to stop there. We could keep going. We get the same idea. So now I'm going to share the screen and we'll start talking about gametogenesis. Okay, so far so good. Okay, so this is a diagram of a human testis. Here is cut it in sagittal section. Here one of the little tubes is cut in cross section. The sperm cells are made in these little tubes here, which we can see as the little light colored structures. They're made in here. They travel up through this network. This is the epididymis, where the sperm cells are stored and where they mature, and then they will eventually pass out of the testis through the vas deferens. This is a cross section. The spermatogenic tissue is right here near the basement membrane of this tissue, and as they divide, the cells migrate towards the lumen. So you'll see spermatogonial cells, primary spermatocytes, secondary spermatocytes, some young immature sperm cells called spermatids, and then finally the sperm cells. Now this is accomplished by meiosis. Here's a very basic diagram of meiosis. This is could be used for male or female both, although there are some differences, which we'll talk about in a few minutes. Meiosis turns diploid cells into haploid cells. Diploid cells have 2 sets of chromosomes. In humans, that it consists of 46 chromosomes or 23 pairs. This diagram is only showing 2 pairs of chromosomes. So here we see one pair and here we see the other pair. Now, when meiosis starts, the first thing that happens is these chromosomes will replicate. So each of these chromosomes we see here will have 2 strands instead of 4 strands. And then after that happens, the chromosomes will undergo a process called synapsis, where the homologous chromosomes will attach to each other, and then they can exchange some genetic material. This process is called crossing over and results in recombination. Recombination is important because it will take some of the alleles off of one chromosome and stick them on the other one. So here we are. We've seen that process. Now, it is still a diploid cell. This one up here has 4 chromosomes, and this one does too. Or this one does also. The way to count the number of chromosomes in a cell is to actually count the centromeres. The centromeres are the little parts of the chromosome that attach to the spindle fibers. So I have 1, 2, 3, 4 centromeres here, and I also have 1, 2, 3, and 4 up here. Okay, so this is still a diploid cell, even though it has twice as much genetic material in it. This diploid cell will then divide in what is called the first meiotic division to produce 2 cells that are now haploid. We now have only 2 centromeres in each cell, so that means that there are 2 chromosomes in each cell. This is called meiosis 1, or the reduction division. It's called meiosis 1 because it's the first division. It's called the reduction division because the chromosome number is reduced from diploid to haploid, in this case going from 4 to 2. Then we do the second meiotic division, which is called meiosis 2. What happens in this case, each of these centromeres replicates, and then the cells split apart again. So we get 4 cells, each of which has 1 chromosome in it. The difference between this cell, which is a gamete, and this one, which is a secondary site cell, is that these have 2 chromatids, or 2 strands to each chromosome, and these do not. They just have 1. So if we compare these cells here, which would be gametes, we have half as many chromosomes as we had in the parent cell. So here's how that happens in the males. In the male, the progenitor or progenitor cells are called spermatogonial cells. These start developing in the male before he's born. It's like in the 3rd month they will start developing, and they will undergo mitosis for many, 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 many generations. In fact, they will be undergoing mitosis until this male reaches puberty. These are 2N cells. At puberty, he will have literally billions of these cells. At that time, about half of them will differentiate and will become a primary spermatocyte. The other half will divide again, and that basically replaces this population. So if you had 2 billion to start with, half of them differentiate, the other 1 billion divide again to produce another 2 million, and then half of those will differentiate. The bottom line is the male never runs out of primary spermatocytes. So in the first division, the primary spermatocytes will replicate the chromosomes. So this stage right here represents this. And then they go through the first meiotic division. Now it is a secondary spermatocyte. Instead of having 2 sets of chromosomes, they now have 1. So these cells here are representing these cells, and then those will again quickly divide, and we get 4 small cells that have 1 set of chromosomes. These are called spermatids, and then they will undergo some differentiation to become sperm or spermatozoa. And the female, it's different. The female starts off with primitive cells called oogonial cells. They also start dividing in the embryo stage, and in fact, all of the oogonial cells that the female will have will be present when she's born. She will have maybe 600 to 800,000 of these things, whereas the male, remember, has a totally limitless number. So these cells here will eventually develop into primary oocytes. So here's a primary oocyte found in the ovary. The primary oocyte will replicate the chromosomes and do the first meiotic division to become a secondary oocyte. but the division happens in a different way. They will produce one large cell, which is the secondary oocyte, and the other cell is a small polar body, which is nothing but a nucleus. So if we scroll back a couple of slides, at this point right here, the cytoplasm is divided unevenly in the female, so we'll get one secondary oocyte that is essentially the same size as the primary. The other cell will be a tiny little cell with nothing but a nucleus in it, and it is called a polar body. So here we see the polar body, and the polar body will disintegrate. Then this cell here, the secondary spermatocyte, is what gets ovulated and comes out of the ovary. It will not do the second meiotic division unless it gets fertilized. If it gets fertilized, which normally would occur in the fallopian tube, then it will do the second meiotic division, produce a second polar body, and then this cell with the other nucleus, which came in from the sperm cell, will be a zygote. So we don't actually have an ovum that is totally separated from the male's sperm cell. In other words, we can't get an ovum if the spermatocyte doesn't get fertilized first. This shows the process of ovulation in a female. Here is the primary follicle, which will develop into a secondary follicle, which will develop into a mature follicle, which will develop into a Graafian follicle. And this is a fluid-filled chamber here. It'll get so big it will rupture and blow a hole in the side of the ovary, and then ovulation will occur. Ovulation is the secondary oocyte, and it will become an ovum only if it gets fertilized. Okay, anyway, this is the last video, and I hope you have a good week, and I will talk to you later.